The UniProt KB database contains individual protein entries that provide detailed information about the protein. At the top of the page you can see the UniProt entry accession and name, the name of the protein and gene, the organism, whether the protein entry has been manually reviewed by a UniProt curator, its annotation score, and the evidence level for its existence. You can use buttons on top of the page to run a blast sequence similarity search on the entry, align it with all its isoforms, view it in various formats, add it to your basket to save for later, or view the history of changes to this entry. You send feedback about it to UniProt by clicking here. On the left-hand side of the page there is a display section which allows you to switch between different views for this entry. You can choose between the default entry view, the feature viewer, and the featured table. The feature viewer allows you to see all sequence features together in a visual manner. Features are arranged into categories such as domains and sites, molecule processing, post-translational modifications, mutagenesis and variants, etc. You can click on a category to expand it further. The ruler on top represents the sequence length of the protein. To see more information about a feature, you can click on it. This will bring up a tooltip with information and also highlight the sequence position of the feature. For example, let's click on this active site feature. To see which other features are co-localized with this active site, let us zoom in further. This can be done by dragging the handles on the ruler or clicking on the zoom button to the left of it. We can now see that our feature coincides with a peak in variance, so we click on the variants to expand the view. We find 3 variants color-coded to indicate involvement with a disease. You can click on them to see more details, including evidence. The variant view maps all variants on a matrix with the sequence position on the x-axis and the possible amino acid changes on the y-axis. All variants are color-coded, going from deep red for disease-related variants from UniProt to a light green for non-disease-related variants from UniProt, and shades in between reflecting variants from large-scale studies like COSMIC, 1000 Genome, ESP and EXAC. You can also filter the variants to see only UniProt data using filters on the left-hand side. Switching back to the default entry view, the left-hand side also provides a navigation bar where you can see all the different categories of information available for this protein, such as function, names and taxonomy, sequence, etc. Any unavailable categories will be greyed out. You can click on the category names to jump straight to them. You can customize your view by deactivating any categories you are not interested in seeing. You may see evidence tags in the content, providing details of the source of this information. The protein entry also contains links to similar proteins that may be of interest, based on the sequence clusters dataset known as UniRef.